Thank you, Heath. Our next speaker is Gili Greenbaum.
Okay, good afternoon, everyone. So I will be talking about population structure or genetic population structure. So what is a population structure? We have here a population. A population, of course, is composed of individuals. And we know that each one of those individuals is carrying genetic information. What we would like to be able to do is to use this genetic information in order to understand how the population is structured. For example, we would like to know if we can partition the population into biologically meaningful groups based only on the genetic information. So several methods have been developed in order to do that, and these methods can be divided broadly into two approaches: model-based approach and a model-free approach. So the model-based approach, for example, as implemented in the program structure, we assume an underlying model and we try to parameterize the model using the observed genetic information. In the model-free approach, we do not assume any underlying model. We try to represent the data in such a way which we can interpret as a population structure. For example, in principal component analysis, we can project the data on 2 or 3 meaningful dimensions and try to use this to understand what the population structure is. These methods, these model-free methods, have several limitations. For example, it's not trivial how to partition the population to groups even after, for example, projection. So I've been working on a model-free approach for inference of population structure, which is based on concepts and ideas from network theory. So just a few words about network theory. Network theory is a branch of discrete mathematics that is concerned with constructs called networks, which are composed of discrete elements, which we call nodes, and these nodes are connected by an array of edges. And in the network theory, we try to study the topologies of these constructs in order to learn about the underlying processes in the systems in question. So there's many applications for network theory, also in genetics, gene regulatory networks, gene flow in patch networks, and how do we use network theory to infer a population structure. So what we will do, we will construct a network where the nodes of the network are the different individuals in the population, or more precisely, the genetic information of those individuals. And for edges, we will characterize the edges with the genetic similarity between the individuals. So if we connect all the individuals by using the genetic similarity measure, one genetic similarity measure, we get this network. And for example, this edge connecting these two individuals is a thick edge. This means these individuals are genetically similar. And these two individuals here are not that similar. They are connected by a thin edge. So you see this structure is rather complex. So in order to simplify it, we can remove some of the weaker edges from the network. So we remove some edges. We remove some more edges. And what you might notice is that these structures emerge within the network of groups of nodes that are more densely connected between themselves than they are to the rest of the network. And we call these dense structures in network, we call them communities in network theory. And this field of research of identifying communities, analyzing communities, is a very active field of research today in network theory. I'll just mention one important concept. The measure of modularity is a quality measure which evaluates how well a network is partitioned into communities. And it does so by comparing the density inside the communities, the intra-community density, versus what you would expect under a null model, which preserves the major topological characteristics of the network. So I wrote a software. It's called NetStruct. It's publicly available. and you supply it with your genotypes for your population. It constructs the network, removes the weaker edges, applies these community detection methods, and gives you, as a result, the groups of closely related individuals in your population. So we wanted to test it out. We tested it out on some simulations. So here you have a very simple model, population of size n, split n generations ago to 3 subpopulations also of size n. And we simulated the coalescent process using the program SymCoal on 5,000 SNPs. And the resulting genotypes we gave as input to NetStruct. So here are the results. The different colors are the different communities detected by NetStruct. And what you can see is that these 3 communities, they perfectly match the simulated populations. If we remove some more edges from the network, we can remove edges. We see the same partition remains until the network breaks up into these small disconnected units and the analysis stops. Okay, this was a pretty simple model. We can complicate it just a little bit by adding some migration between population 1 and 2 after the split. So now we would expect that on one level we want to see the 3 different subpopulations, but on another level we also want to see that populations 1 and 2 are more similar than they are to population 3. Again, we ran the coalescent process. The genotypes were supplied as input to NetStruct. And this is the result. So here we get only 2 communities, the red and the blue. The red corresponding to populations 1 and 2, and the blue corresponding to population 3. Now we start removing edges from this network. We remove edges. We see the same partition remains until one point when a third subpopulation is detected, and this remains until the network breaks up. So what we see with this simulation is that we can detect the basic population structure, but also using this process of removing edges and applying community detection methods, we can detect the different levels of hierarchy of structure in the population. Next, we wanted to test it on empirical data. So we took some, we extracted data from the HapMap project. This data includes 11 subpopulations, human subpopulations. We randomly selected 50 individuals from each population and 1,000 SNPs from each chromosome. And this was run with NetStruct. So this is the result. For the lower threshold, we get a partition to 2 communities which correspond to African and non-African populations. With the medium threshold, the edge removal threshold, we get 3 communities corresponding to Africa, Indo-Europe, and East Asia. And with the highest threshold, we get 6 communities just from the genetic information corresponding to Africa, Europe, Mexico, India, China, and Japan. So we can see that we can uncover the known human population structure just based on the genetic information. And we also get the different levels of hierarchy, which of course correspond to the major migration events in human history. So this analysis try to look at groups of related individuals, but I was also interested in looking at how specific individuals are connected in these kind of networks. So I want you to pay attention to this individual here. This individual here is assigned to the red community. But if we look at the edges connecting this individual, we see that this individual is connected only to individuals within its community, within the red community. So we would like to say that this individual is very strongly associated with its assigned community. On the other hand, if you look at this individual is also assigned to the red community. But if we look at the edges connecting this individual, we see that it's also connected to individuals in other community. So this would mean that this individual is more weakly associated with the red community. So in order to quantify this notion, we developed a network measure, which we call the strength of association. The strength of association is based on looking at the modularity of the network versus alternative modularities where the individual is assigned to other communities. So what can we do with this measure of strength of association? We did some simulations, and we saw that we can use it to identify hybrid individuals, first and second generation hybrid individuals, as individuals with very low strength of association, because they are between communities. But I will show here another analysis, which we call the strength of association distribution analysis, or SAD analysis. So here we see the partition of the HapMap data to 3 communities, African, Indo-European, and East Asian. And below, what you see is the distribution of the strength of association of the individuals in the different communities. And you can see that these are very different distributions. For example, the African distribution is very narrow and with a high mean. This means that this group is very cohesive genetically. Individuals are very much associated with this group, compared, for example, with the Indo-European group, which has a lower mean and a higher dispersion, meaning that individuals, or at least some individuals, are less associated with this group. We can look a little bit in more detail, for example, at the African community. We see we have a bimodal distribution. So what we can do now is we can decompose it back into the sampled subpopulations from the HapMap data. And we can see that the higher mode, the individuals most associated with this group, are from Nigerian and Kenyan descent, while the lower mode is due to individuals from African American and Maasai descent. So we interpret this as that these two groups have experienced more gene flow in the recent past. We can do the same exercise with the Indo-European community. It has a high mode and a very long left tail. We can see that the mode corresponds to individuals from European ancestry, while individuals from India and Mexico are less associated with this Indo-European group. So just to summarize, we saw that we can use these genetic similarity networks to infer population structure. This has several advantages. This is a model-free network. We do not assume any underlying model. We can detect structure at different hierarchical levels. We also have significant tests in order to verify this. And we have other analyses, like the SAD analysis, to explore other aspects of population structure, for example, the gene flow patterns. And I believe that network theory has a lot more to offer to population genetics, and I'm working on adapting and adopting methods from network theory to answer questions in population genetics. So I encourage you to use NetStruct, and I want to thank all these people and you for listening. I'll be happy to take questions.
Hi, I was wondering whether, so you mentioned that this type of approach allows you to infer hybrid individuals, as you call them, that are first and second generation admixtures. I was wondering whether you could use it to infer earlier historic admixture events, and how this method compares to other methods that are used to do that, like D and F statistics.
Yeah. So, the hybridization simulations, we looked if we can identify specific individuals. This is something important in ecological studies, to see if you can identify the specific individuals, which is a hybrid. This is difficult to take back more than a few generations, and actually you can see here the results from the hybridization. You can see that once gene flow between populations is too high, the second generation individuals in red, these are the individuals in red, are no longer detected as outliers. So there is a limitation to how far back you can do that. We have compared it with other methods as well. You can see it in the paper or discuss later. Very quick.
Yeah, what are the primary advantages over PCA?
Okay, that's a long question, actually. Generally speaking, in PCA, you don't have a partition, an easy way to partition it. You can use analysis like k-means and other approaches in order to partition it into groups, but it's not trivial, and the theory there is not that developed.
Thank you, Gili.